In this section, we will talk about binding motif, immunoproteasome, immune escape, central and peripheral tolerance. First, we will discuss about the antigen processing and presentation of endogenous antigen. MHC class I presented peptides derive from proteins that are present in the cytoplasm. This allows the immune system to recognize cells that have been infected by viruses or by intracellular bacteria. Theoretically, this also allows recognition of mutated proteins of cancer cells. In order to preserve cell viability, damaged, denatured, unassembled, or misfolded proteins must be eliminated. Normal proteins also need to be constantly degraded and replaced with half-lives, ranging from a few minutes to several days or weeks. Proteins are tagged for destruction by the protein ubiquitin. Multi-ubiquitinated proteins are recognized by the proteasome. Proteasome is a large protease complex with a molecular weight of 2,000 kilodalton. It is barrel-shaped and made of a core unit of 20S, which is 650 kilodalton. It also has two additional outer rings of 19S, each is 700 kilodalton in size. The four rings of the 20S proteasome consist of two inner, beta units and two alpha units, all of them consisting of 7 different subunits, which surround a central chamber, where proteolysis occurs. The majority of peptides processed by the proteasome are quickly degraded into single amino acids by cytosolic or nuclear enzymes. Only a few peptides, mostly with a size of 8 to 10 amino acids, are transported into the endoplasmic reticulum by a heterodimer or protein complex called transporter associated with antigen processing, or TAP. In endoplasmic reticulum, MHC is unfolded initially. It is then folded by calreticulin. In the endoplasmic reticulum, peptides bind to the peptide binding groove of MHC class I proteins. If the side chains of their amino acids fit properly with the amino acids of the binding groove of the MHC1 alpha chain. Initially, the alpha chain is bound by chaperone proteins such as calnexin, calreticulin, ERP57, which regulate peptide binding. Tapicin bridges the alpha chain to TAP. This MHC peptide complex moves through Golgi apparatus to cell surface. Now, let's discuss the binding motif. This is alpha chain of MHC class I. 2 or 3 pockets in each MHC molecule, except only particular amino acids, for example, aliphatic amino acids or basic amino acids. These are called anchor residues. They differ for different MHC1 alleles, since each allele has a different sequence of the alpha chain. In most cases, the amino acids in position 2 and 9 are crucial for binding. So, amino acids at position 2 and 9 is anchor residue. Anchor residues of peptides that bind with high affinity to 3 frequent HLA alleles are leucine at position 2 and valine or tyrosine at position 9 are anchor residues of peptides which bind with high affinity to HLA A0201. Similarly, leucine at position 2 and lysine at position 9 are anchor residues of peptides which bind with high affinity to HLA A3. Similarly, leucine at position 2 and proline at position 9 are anchor residues of peptides which bind with high affinity to HLA-B7. Peptides with a different amino acid in these positions may bind, but with a lower affinity. The restrictions dictated by the anchor residues are responsible for the fact that out of a whole protein, only a small number of peptides bind with high affinity to a particular HLA allele. However, since each individual may have up to 6 different HLA alleles, 3 maternal and 3 paternal alleles of HLA A, B, and C, a broad spectrum of peptides can still be presented. Computer programs available through the internet can predict binding epitopes of a given protein for the most common HLA haplotypes. Do you know what is immunoproteasome? Well, lymphoid tissue and cells exposed to interferon gamma, so they have a slightly different proteasome complex as some of the beta subunits are substituted by subunits with different substrate preferences, such as LMP2, LMP7, and MECL1. The proteolytic activity of the immunoproteasome appears to be enhanced compared with the constitutive proteasome, which is in accordance with the higher antigen processing requirements of antigen-presenting cells and of virus-infected cells exposed to interferon gamma. Proteins processed by the immunoproteasome can generate a whole different set of peptides. Then the proteins not processed by immunoproteasome, or the protein processed by simple proteasome. As you can see, the sequence of peptides generated by immune proteasome is different from the sequence of peptide generated by proteasome. Now, let's discuss the immune escape mechanism. T cell recognition can be prevented by immune escape mechanisms. As we have studied that virus after entering the cell, as this is a virus-infected or tumor cell, the antigen is tagged by ubiquitin. It then passes the proteasome and converts to peptide and enters the endoplasmic reticulum through TAP. So defects, for example, mutations in the sequence of TAP proteins can prevent the transport of peptides into the endoplasmic reticulum, so that empty MHC1 molecules are generated. It passes Golgi apparatus. MHC1 are unstable and dissociate quickly from the cell membrane. Viral proteins, such as the herpes virus proteins, US2 and US11, can induce active export of MHC molecules from the endoplasmic reticulum, preventing loading of viral peptides in the MHC molecules. Indeed, low levels of MHC1 are frequently found in viral infections. Now let's have an overview of central and peripheral tolerance. Since many endogenous self-peptides are presented by MHC1 molecules to T cells, two major mechanisms exist to prevent autoreactivity. First is central tolerance. In the thymus, presentation of self-antigens by thymic epithelia or dendritic cells to thymocytes, it can lead to clonal deletion of antigen-specific T cells, called as central tolerance by clonal deletion. In the periphery tolerance, recognition of self-antigens by antigen-specific T cells that may have survived thymic selection leads to abortive activation or apoptosis or clonal anergy because of a lack of co-stimulatory molecules, called as peripheral tolerance. This was all about today's section on binding motif, immunoproteasome, immune escape, central and peripheral tolerance. In our next section, we will talk about anchor amino acid, allele-specific motifs, and T cell activation.